Hi everyone, today we'll be talking about practical experiments, more specifically experiments on plants. Okay, so I'm trying to cover as many plant experiments as I can. So to start off, we have photosynthesis experiments. Now I'm going to write down the photosynthesis equation over here. And why we are doing this is because I want us to focus on the independent variables. Alright? Now, what are independent variables? They are the factors we manipulate. So as an experimenter, what am I changing about the experiment so that I can investigate something? Okay, so what am I changing about the setup? So for photosynthesis, it can be things like carbon dioxide, I can change light intensity, I can also change chlorophyll. For carbon dioxide concentration, I can actually change it by altering the concentration of sodium hydrogen carbonate solution. Okay, of course this is for aquatic plants only. For light intensity, I can change it by varying the distance of the lamp from the plant, and I can also switch out the light bulb for a light bulb with a higher wattage or a higher power. Okay, so it's actually brighter. I can also change the chlorophyll concentration by changing the concentration of the leaf solution that is being prepared. Okay, now I know some of you might be thinking, we didn't really learn how to change this, but this is what happened for last year's 2019 end of year practical. So basically they were given different chlorophyll suspension, and then they had to investigate how the concentration of chlorophyll affected photosynthesis. So that is something that we can also consider when we are preparing for experiments. Next, we can vary the temperature by changing the water bath, so by either adding hot water or adding ice. And lastly, you can change pH by changing the acid or alkali added to the sodium hydrogen carbonate solution. So all of these things are independent variables because when I change them in an experiment, they will cause a change in the rate of photosynthesis. Okay, so how do I measure this? It is by measuring my dependent variable. So basically, dependent variables are results that we can measure, or the data that we collect, or the data that we observe. And dependent variables change when the independent variable changes. Okay, so normally in photosynthesis experiments, we will try to measure oxygen and glucose to give us a gauge of what is the photosynthesis rate. Okay, so photosynthesis rate will be my dependent variable. So for example, I can measure the rate of oxygen production. So this will be my dependent variable. How can I do so? I can do by counting the number of bubbles produced over time. Okay, classic example. Or I can also measure the volume of oxygen produced by using a gas syringe. This will be more accurate than counting bubbles. Now, apart from counting bubbles, what I can do is that I can actually measure the distance moved by the meniscus. Okay, so we have done this experiment before. So remember, you've marked out on like a glass tube where the water originally was, and then after 20 minutes, where is the new water level? Now, the water level changes because carbon dioxide is absorbed and used from the solution. So the volume of solution actually changes. It becomes less. So there are a number of ways of setting up the experiment. So you can see the one on the left is something that we have done before. So in this case, the meniscus will move upwards, while the one on the right, the meniscus will move towards the left. Okay, but the idea is generally the same. The third way of measuring rate of oxygen production is by using a leaf disc method. Okay, so what I'm doing is I am measuring the time taken for leaf discs to float. So what happens is that I will actually punch some holes from a leaf, and I will suck out the air from the leaves by using a vacuum, and then I will place it into a sodium hydrogen carbonate solution. They will actually sink, and after a while they will start to float because during photosynthesis, oxygen is accumulated in the leaf disc itself, and this causes the leaf disc to float up after some time because air is less dense, and then it will eventually float up. So the time taken can actually be used as a gauge of the oxygen production as well. Okay, So all of these methods, they are either measuring production of oxygen or carbon dioxide absorption. And there is one last one that I want to touch upon, is by measuring the time taken for color change of an indicator. Now we know that carbon dioxide is actually acidic and it is being absorbed. So what we would expect is after a while, right, the carbon dioxide will be used up and it becomes less acidic. So if I have an indicator that is inside the sodium hydrogen carbonate solution, it should change color when carbon dioxide is being used up. So we have talked about how oxygen production can tell us about the rate of photosynthesis. Next, we can actually talk about how glucose can also tell us whether photosynthesis has occurred or not by actually measuring the starch that is being produced. Now, unfortunately, this method is usually a yes or no kind of thing. So it can tell us photosynthesis has occurred or it has not occurred, but it does not tell us about the rate because we don't know how much starch is being produced. It's either yes, present, or absent. Unless we actually measure the area of the leaf with starch. And usually for this kind of things, we only do it on variegated leaves, which are these leaves with some parts that are green, some parts are white, and we do it to measure the effect of chlorophyll on photosynthesis. Okay, but just a sidetrack, how to measure area of a leaf. If you remember lower sec, the good old counting squares method. So you get transparency with a grid and then you just count the number of squares. Now the more important thing about the starch test is what is the importance of each step. Okay, so boiling, it helps to denature the photosynthetic enzymes. So we know that it has stopped once I start. Okay, alcohol, it actually helps to decolorize the leaf so that I can see the color change more obviously without the green color messing around. The hot water is to soften the leaf so that iodine can be absorbed. And lastly, iodine is for us to observe the color change from brown to blue-black if this is a positive test, or remains brown if it's a negative test. Okay, please in your answer do not write down remains the same because it does not tell us any result at all. What do you mean by the same? Okay, there is no like original color, there is no end color. It doesn't really tell us any data at all. Okay, so try to write down the color and be specific. Moving on to transpiration experiments in plants. So we want to find out the rate of transpiration. Okay, and if you recall from our transport in plants, the factors that are affecting rate of transpiration, my independent variables will be things like wind speed, which I can actually alter by changing fan speed. Light intensity, which same thing, change distance of the lamp from the plant. Humidity, which is actually kind of the same thing as wind, so less wind equals more humidity. And lastly, temperature, which is very hard to manipulate inside a lab. Okay, so a new thing that I want to talk about is stomata density. So basically, how many stomata is there, on the upper versus lower surface of the leaf. And we can actually change this by using Vaseline, which is a waterproof substance, to prevent water from leaving the leaf. So we can actually compare transpiration on different surfaces of the leaf. And the dependent variables in transpiration, or how can we measure the rate of transpiration, which is the rate of water loss. We can measure the rate of water uptake. So this might seem a bit like, what? You're measuring rate of water loss by measuring rate of water uptake. But remember, transpiration, right, when I lose water, it actually helps to draw water up. So the rate of water loss is actually equal to the rate of water uptake in a plant. And remember, most of the water is lost anyway. I only use 1% for photosynthesis. So this is a pretty good gauge for transpiration. Another way of measuring transpiration is the mass of water that is lost, right? So the first thing that I can do is I can weigh the leaf before and after the condition. So I weigh the leaf on an electronic balance, I hang it up on a retort stand, maybe I expose it to a little bit of wind, and then afterwards I go and measure the leaf again to see how much water has been lost. And this will give us a pretty good idea about the rate of transpiration that has occurred in the plant. We can also measure water level change over time. Now, when we look at this setup, water is lost from this sprig of leaves, and this actually causes the water level in this calibrated pipette to fall over time. So we can actually measure the volume of water lost through transpiration. Okay, so imagine this whole setup is actually clamped to a retort stand, not just floating around. The third method that we can actually use to measure transpiration rate is by using a potometer. Okay, so I think we have seen this before in theory. So what happens is that same thing, water is being lost from the leaves of the plant, and this causes the air bubble to move towards the left side. Alright, and we can actually use a ruler to measure the distance moved by the bubble across time. Now, some key things to note is that you must have an airtight seal, and this reservoir is actually to help us to reset the bubble. So I can open the tap and then the bubble will move back to the right side. Finally, let's talk about sources of error. Okay, this is a super common question. Now, when we talk about source of error, we are not asking about errors that are human in nature or that are preventable normally. Okay, because you should have the practical skills required to actually prevent all these kind of mistakes from happening in the first place. Okay, so things will be like parallax error. These are things that can be already prevented, so you should not be writing these things down. So the first kind of error will be precision of instruments. So is there a more precise tool to use? So when we compare plastic droppers to measuring cylinders and syringes, we all know that syringe is definitely more accurate. Okay, but it really depends on the volume that we are wanting to measure. So for example, if I'm measuring something that is 2 cm cube, then using a syringe will be more appropriate because it is best for volumes that are below 5 cm cube or even 10 cm cube because it has more divisions. That means there is more markings and it's more accurate. But if I want to measure a larger volume, I will try to use a measuring cylinder instead. Okay, so it depends on what the question wants you to measure. Another thing that we can talk about is analog clock versus digital stopwatch. Okay, so it depends again on what we want to measure. If we are measuring incubation period, meaning measure 20 minutes for the whole experiment to run, then analog clock would be good enough. But if I am measuring a particular event, such as a color change, time taken for color change, then a digital stopwatch will be better because I can even stop it down to the milliseconds. So you have to take a look at the tools and equipment that you are being provided in the experiment and decide if that is a source of error. The second kind is biological specimens. So are we comparing leaves of the same plant? Now, it is best if you are actually given leaves of the same plant, but more often than not, we never know, right, whether hydrilla is from the same plant or not. Why do we want it to be from the same plant? It's because the plant would have received the same kind of nutrients, same amount of nutrients, it's been watered the same number of times, exposed to the same kind of sunlight. And this will actually help us to remove any other factors that might have affected the results. Now, many students actually confuse this part with control variables. Control variables will actually refer to the same length or mass of the plant. So for example, did you measure like 3 cm of hydrilla or 5 grams of hydrilla? And usually in the practical exam itself, they would already tell you how much to measure. So that is actually a control variable. That means things that we want to keep constant. Okay, so do not confuse this with source of error in terms of biological specimens. So usually when we ask for source of error, these two errors, they will not be like the first that I would think of. Okay, so precision of instruments really depends case to case basis because more often than not, we will already give you the best instruments. Biological specimens in this case, not the best. Alright, so let me talk about other things that we can write in your answer. So things like experimental procedure itself, it will be a great source of error. Now, an example will be time for acclimatization. So it takes time for a plant to get used to the environment and start photosynthesis, right? You cannot just dump the plant into the solution and then expect it to be 100% photosynthesis. So normally we'll try to give it about like 5 minutes to get used to the temperature, get used to the solution, and for photosynthesis to actually kickstart before we start counting like number of bubbles or measuring the volume of oxygen being produced. So if they did not write this down, or if they did not give this time in the instructions, then you can write down as an experimental error. Another error will be like counting bubbles, where volume of bubbles may differ. Some might be bigger, some might be smaller, so we can use a gas syringe instead. And when we say color change is relative, so this is more often than not for indicators. So what we can use is a colorimeter or a standard to make sure that it is less subjective and more objective. So if you remember, there was this experiment we did on the yeast where they asked you to breathe into like a test tube of indicator first, and then it changed color, and then you are supposed to use that as a comparison to the rest of the experiment. So that is what we mean by using a color standard to make sure that the color change is all the same. Finally, we can talk about external environment as a source of error. Okay, so there are things in the lab that you cannot change. So example would be like in the photosynthesis experiment, there will be other sources of light, right? Or transpiration, there's other sources of wind. And what we can do then is to actually conduct it in a dark room or conduct it in an airtight container to remove the effects of this external environment. Another thing we cannot control is the heat from the lamp, which might actually increase photosynthesis. So imagine you're doing an experiment on light intensity and your lamp is pretty close to the plant and it's actually emitting some heat, causing the temperature of the solution or the plant to actually increase, and the enzymes are more active. So that might be something that affects the result. So what we can do is use a heat screen instead to shield the heat from reaching the plant. So for questions about source of error, usually it's two parts. They will first ask you what is the source of error, and then the second part will be asking you to suggest how to improve on it. So sources of error I will highlight in red, for example, your clock and the dropper. And to improve, it can be using more accurate instruments that I've highlighted in green. Source of error can be the plant is not used to the environment yet. How we can improve is by adding a 5-minute time for acclimatization to occur before we start experiment. It can be bubbles might differ in size, and we can improve it using a gas syringe. Or color change being relative, so we can improve it by using a colorimeter or a standard. Other sources of wind and light, and we can improve it by removing this from the environment. And lastly, heat from the lamp can be improved by using a heat screen. Okay, so I hope this gives you a better overview of the large number of experiments that can actually occur, and also how we can better answer some of the questions about practicals. Now, do take note that this list is also not exhaustive. Okay, Cambridge may always come up with more interesting experiments, but I hope that this gives you at least some kind of exposure to the kind of setups that they might have, and you realize that most of the time it's always like you measuring the same kind of thing lah, but with just different instruments. Okay, so do try to take note about the fundamental concepts behind all the practical experiments.